Yes, Prash, you're welcome to give us a talk today at the Extra Lab seminars. And today we have our guest from India, and he will present his data on the prostate cancer. And Prash, you're welcome.
Thank you. Thank you so much, Shira, for your kind invitation amid these COVID times. I've been following your talks, and it's been absolutely very useful, for me as well. So I'm currently a senior scientist at Birla Institute of Scientific Research in Jaipur, which is in Northwest India. I obtained my PhD from Aalborg University, Denmark, where I finished my PhD between 2004 and 2009. And subsequently, I was a postdoctoral fellow across 4 different labs in Denmark and US before I moved to India in 2016, where I set up my, the systems genomics group. So today's talk, I would focus on cancer prostate and the kind of rationale behind this particular project. Where are we heading towards cancer prostate in India? The kind of challenges and problems. And as a pilot, we ran whole exome sequencing for cancer prostate cohort from India. And we'll discuss also some of the inherent pathways and the comorbidities associated with these particular patients. And the second and third part of my particular talk would largely delve on the long non-coding RNAs, which our lab is very much interested. We just started, working towards this. And the kind of panel that we wanted to develop as a part of this particular project, I'll discuss certainly. So, as I said, I come from Jaipur. Ours is a not-for-profit research institute called Birla Institute of Scientific Research, which is, two of the leading philanthropic research institutes that India has seen a big uprise over the last 75 years of our independence. That is, the other one is Tata, and the second one is Birla. And our current chairman is C.K. Birla, who is sitting somewhere in the down figure, this one. And we have got state-of-the-art laboratories with 2 main divisions. One is the Biotechnology and Bioinformatics, and the third one is which we call it as the remote sensing, where we collaborate with these biophysicists in procuring some samples from remote areas where we collect the flora and fauna. And we have got a biggest auditorium here. It's quite an amazing place. If you are in India, please feel free to contact me. And we have got a planetarium as well, which is only the third of its planetarium in India. So to start with cancer prostate, so how did this all set? I was doing my postdoc, my third postdoc between 2013 and 2014 from Haja Kadamadine's lab, which is a quantitative genetics lab in Denmark. And since it was a kind of an animal genetics lab, my PI group, they were largely working on the feed efficiency traits. So they would probably feed the animals or overfeed the animals, and they would ask, what would happen to those particular animals, if they're really overfed. So intuitively, so if we eat normally, we think that, we are probably healthy, that's the bottom line. But if we really eat less, that is, people might really become unhealthy. But the most important thing would be if we really eat optimally across different time periods, that would indicate that, we are again, the so-called, the healthy cohort. But if you really eat too much across irregular intervals, that's where this unhealthiness or the so-called obesity or cancer prostate, would really come into picture. So that was what, we were really asking, looking into some of the important animals where, we were working on, we were working with, like all the eutherians, like the cows, the so-called the pigs, and even the mice. So the animals would not only become obese, but they would also become cancer prostate victims. Okay, so this was one of those particular things. And then when I set up my particular group in India, we asked if there are any cancer prostate victims in India. Surprisingly, as you pretty well know, not only from the northwestern state of Rajasthan that I come from, but all over India, many Indians are vegetarians. So over 63% of Indian population are vegetarians. And we found that there are no prostate cancer specific mutations known. So this was also one of those particular reasons why one of these particular projects is really funded. So the very first part of my talk would delve on the pilot study on the mutations, ascertaining the mutations relevant to Indian population. So if you really look into the basics here, so most important stuff here is over 1 in 9 men would probably be diagnosed with cancer prostate during his lifetime. So this is also seen by and large in Europe as well as American cohort, and as well as the African and Han Chinese, by and large. And it is the second leading cause of death globally, and it's the third most prevalent cancer known in India. And as a result, there is a growing geriatric population where there could be declining fertility rates, and as a result, this cancer prostate incidence is really upcoming by and large. So if you really look into the very basic problem, why and how or wherewithal this cancer prostate victims could be could be used. So as you could see here, the African American population are more prone to be cancer prostate victims. That's because they eat a lot of red meat. So certainly the non-vegetarian dietary style is certainly one of those particular causal reasons for cancer prostate. Whereas many Asian Indians, typically South Asian Indians, they eat a lot of vegetarian diet, so probably they're not really prone to that. And as I could see, from this particular incidence, there has been an absolute mortality rate that has been known in Indian cohort as well, not only just in US. There are different consortia groups where research is burgeoning with respect to these particular cancer prostate incidents, and as well as the mortality. A large number of cell lines have already been developed, but in India, there are no such particular studies. There is a population-based cancer registry, which we call it as a Globocon statistics. So as far as 2018 and 2020, the latest is 2020, which was released just a couple of days ago, which I couldn't update it. There have been rising number of cases over here in India as well, as you could see here, and the 5-year prevalence is about 47,558. So if you really see, there could be as many good number of risk factors. So there is an association between fat consumption, and there are also countries with greater per capita milk consumption, although it is assumed that vitamin D could also play a very important role, because vitamin D is an absolute milk is an absolute source for vitamin D as well. But if you really see, there has been no risk of association between smoking and as well as alcohol consumption. Although there have been some reports, one or two, but they have not really been subjected to this. But if you really see into the economic conditions, we could vehemently say that if there is a low per capita income, okay, there could be likely chance that the cancer prostate is absolutely less. Whereas if there is a high per capita income, there could be a cancer prostate risk. So the best example is, of course, the US and the European states. So even if you really compare the US and European cohort with the African American population or even the African population, the cancer prostate risk is relatively less. So from the pathological perspective, from the pathodiagnostics, there are a good number of tests that are done. The very basic test is the digital rectal examination. There are, of course, the kallikrein markers, the human kallikrein markers, which they are called as HK3. And of course, the TNM reading is done. The biopsies are probably checked. But the most important grading is, which we call it as the Gleason score. So the Gleason score is given, between 1 and 10. So if there is a score between 1 and 5, they're all the indolent tumors. They are usually the benign, which we call it as a benign prostatic hyperplasia. And if there are between 6, 7, and 8, between 6 and 8, they are what we call it as intermediate Gleason scores, which means they're malignant, but with intermediate prevalence of tumor. And if there is 9 and 10, they are absolutely high risk. Recently, if you really see over the last 4 or 5 years or so, there have been a good number of biomarkers that have been studied. One of those particular well-known biomarkers, and which is one of my favorites, that is associated with long non-coding RNA, is MALAT1, which is a well-known urinary biomarker that has been characterized across all genitourinary cancers, including cancer prostate. So as a part, our first ethics clearance, which we have taken way back in 2018, was with a cohort of 120. But it so happened that our clinical facility is about 10 kilometers away. And there is an inherent stigma in getting the samples as well, although there are many cancer prostate victims in India. So if there is a scrotal pain or there's a pain in the rectum, the men wouldn't even show up to the hospitals. That has been a kind of big concern. So even if there are cancer prostate victims as well, many of them wouldn't even reach the hospital, even reach the clinic for the first aid or even for the first pathodiagnostics. That has been a major concern. So getting samples has been a kind of an inherent concern. So as a result, we had the very first cohort of small set of 10 samples with these particular characteristic features: 5 benign prostatic hyperplasia, which are the indolent, and 2 with intermediate Gleason scores, and 3 with more than 7, which is 8, 9, and 10. Although 8 is intermediate, it is still considered as a high-grade tumor, and all of them were all the FFP tissue blocks. The ethics clearance was judiciously taken from our clinic, and this is the inclusion criteria that we had for our case. So we had, of course, cancer prostate is seen only in age relatively above 55. The youngest patient whom we have seen was about 53, as we could see. Although there were some cases, above 45 as well, because the prostate, wouldn't enlarge, by that particular time. So these are all the control cases, and now we have already finished another 70 exomes as well. As a systems biologist, we have benchmarked this particular pipeline. So where we start with a raw FASTQ reads. So post whole exome sequencing, you obtain what we call it as the FASTQ reads. So once FASTQ reads are given as a kind of an input, our pipeline will give you the number of SNPs in the BCF file, where we will know whether or not any SNPs are pathogenic to our native condition or not. So this is our benchmark pipeline, which has been downloaded or used, at least 25,000 times. This was a paper, which we have published. And then this is the inherent samples across the Gleason scores, which I was just telling you, across the primary and as well as the secondary Gleason scores. But interestingly, all the 10 samples which we had, many of them are vegetarians. When I say many of them, in fact, all of them are vegetarians. There's only one person who would have egg and as well as chicken. When we had this informed consent, taken. Okay, and interestingly, all of them were really, cancer prostate victims. So that was a kind of a question, we really asked. So from our results, as you could see, this is in a whole exome sequencing, the most important thing is the depth. So which we call it as the depth of a coverage. So the ideal depth, which we usually take is 20, but between 5 and 20 are always good, or about 20 is absolutely great. So we have classified all our samples across these particular things where we have considered more than 5 and less than 20 across those particular things. As you could see across the cases, we have found number of variants which are seen in the blue circle of the Venn. And the other one is the yellow one shows the control or the indolent tumors. So if you really see, relatively, we found a good number of variants where the quality check was passed, and we have also subjected these particular variants for downstream validation. But if you really see the take-home message from this, this is pretty interesting here. So all the circular ones, as these are all the Indian, variants or the genes that have been responsible by and large. Whereas the square ones are all those ones with Caucasian and as well as the West. So as you could see, like any other case, BRCA2 plays a very important role, for tumorigenesis or for pathogenesis. There are also some other variants across the genes, like GNP tab, and of course the tumor suppressor TP53. But the most important things which we really found was something related to obesity, which is the Western population. There are also some of these particular genes that are related to the mitochondrial, dysfunction or oxidative phosphorylation. That if a person who is cancer prostate victim, okay, he has, say, he is vitamin D deficient probably, and he would probably be obese as well, from the informed consent and the clinical phenotype, conditions which we have indicated. So we have found, some new markers or new particular variants that I have probably not seen in Caucasian or the Western population. So this was a simple or a very basic pilot study which we published. And another work, with respect to the differential gene expression is in the pipeline. Okay, so a particular talk, we conclude this way that there are patients, who are which prostate victims. And our pilot study has given a kind of a efficiency. We also found variants that are associated with PUSH2A and P53. Although the whole exome sequencing pipeline, does not detect any copy number variants, Yeah, hello. Can you hear me, please? Yeah.
Yeah, we hear you. Please continue.
Yeah. Yeah. Thank you. Yeah. I'm sorry. I'm just sharing the screen. Could you hear my first part of my talk, Ira? I'm
Sure.
really
Yeah,
sorry
sure.
for this
Yeah.
brief connection. Yeah. Yeah. Thank you. So, and we also found these OPA1, which is responsible for the diet-induced obesity, and of course, as I told you, the vitamin D deficiency, CTN, a kind of a basis or a conceptual framework of these particular mutations. Your whole exome sequencing samples from which we are trying to do, it would really form a kind of a basis. So the second part of my particular talk and would largely emphasize genes regulatory. So this is what we are very much interested in our lab. So I've been working on long non-coding RNAs because they play a very important role, and we have developed some kind of strategies to understand the long non-coding RNA protein interactions from the systems biology perspective. So this is what this particular talk would do. So in this particular part, so the most important thing is we know that BRCA2 is a well-known gene responsible for cancer prostate, in addition to the other cancers as well. But we know where our variants are sitting, at least from the Indian perspective. Say for example, this one, which is really marked in circle over here. So what we really asked is if any of these particular neighboring regions of these particular variants harbors any enhancers or any CCF motifs, or if the variant is really sitting within the intergenic framework, are there any long non-coding RNAs that might be juxtaposed or that might be sitting next to that? Okay, so that is one of the important things that we asked. So by their presence, it is believed that, even it is known that the tumorigenesis might be elevated. So there is an elevated risk of pathogenesis if that particular variant is really surrounded by these CTCF motifs or the so-called enhancers. So that also plays whether or not they play a very important role. So that is what we call by genome walking. So we really look into that particular variant. So this is, say, from your window, this is your variant. We walk further upstream 5' UTR and see for these particular CDCF motifs. And of course, we also perform a simplest systems genomic perspective. So we try to blast those particular regions, which is a human long non-coding RNA database. And then we ask if any of these particular long non-coding RNAs play a very important role, in inferring the pathogenesis with that. So this is what we have done across those particular samples. So we have just extracted RNA from the yield is so good with respect to the absorbance and as well as the RNA integrity checks. And then some of them are long non-coding RNAs and some of them are also the long intergenic non-coding RNAs, which we call it as LncRNAs. So among those long non-coding RNAs, so as you could see across those two Excel worksheets, the left or the column B over here indicates the long non-coding RNAs or the PVRNAs. PVRNAs are a family of small non-coding RNAs, just like the microRNAs. They're anywhere between 25 to 40 bases in length. They're not that conserved like the microRNAs, but they play a very important role in what you call regulation. So these are these are PI RNAs and as well as the long non-coding RNAs which we have found, and we used a simplest link pro tool, to infer whether or not this long non-coding RNA proteins are physically interacting with each other. If they stand any chance, to be interacting or not. Okay. So when I say by interaction, it's only by in silico approach. Of course, our clip-seq method or the cross-linking immunoprecipitation or a simplest Western blot, we are trying to perform this, by next month, based on our transcriptome results, that we are trying to get it. So in the meantime, this COVID happened 2020 February. We were in the middle of this particular work, and like any other labs, our lab is also affected. But in between, with the University of Alabama, Keshav Singh's lab and Yek Chen's lab, we came up with a very exciting hypothesis, something related to SARS-CoV-2. So what we observed was, as you pretty well know, as soon as SARS-CoV-2 enters the host, there are two important receptors, ACE2 and the transmembrane protein RSS2. And of course, there are some other interleukins like interleukin 17A, which is also sitting as a SARS-CoV-2. And as a result of this, many of these particular mitochondrial players, they probably, get dysfunctional. And as a result, if in a healthy cell, if there are about 2000 mitochondria per cell, they keep dying. And as a result, the cell dies. And as a result, there is an asphyxiation. And as a result, the person dies. But interim, what we came up recently was, we asked if there are any long non-coding RNAs that are probably known to play a divisive role in this particular pathogenesis. So where those long non-coding RNAs, which are presumptively expressed in cancer prostate population like this, from the tissue expression that we have known from the GTEx database and all those kind of things. And as a result, these long non-coding RNAs are absolutely more stimulative. They're not really that receptive when compared to the other non-prostate victims, So we asked whether or not these really non-coding RNA players, especially the long non-coding RNA players, are really transgressed within the cell. Okay, and this is what we came up with this particular hypothesis. Although this was a mitochondrial hijack paper, which we published in American Journal of Cell Physiology. Our main hypothesis is to look into these long non-coding RNA, players. So the part 2 of my particular talk is, there are some pvRNAs. pvRNAs are anywhere between 21 to 40 nucleotides. Ideally, they are between 21 to 35 nucleotides. We also found some pvRNAs and as well as long non-coding RNAs that play a very important role in regulation. These are those particular candidates, if at all, if we could really identify them from our in silico approach, which we are really validating them in the lab, this indicates that they could be the potential candidates for development of biomarkers, because they are not only playing a very important role in regulation, but they are also aiding an important aspect of pathogenesis. But please bear it in mind, the study so far, which I've described for the first two parts, was taken from the whole exome study. But the whole transcriptome study, which we have the raw reads that are being obtained right now, we are trying to ask if any of these particular genes are differentially expressed, if not constitutively expressed. So if they are differentially expressed, then we can definitely say that maybe the genes that are differentially expressed could also harbor these particular mutations that are responsible for cancer prostate or promulgating the pathogenesis in prostate cancer. So this is where we are just trying to come up with this. So in the meantime, over the last 1 year, in fact, we have been not getting any samples from our clinic. The unfortunate reason is this particular COVID. But as I told you, this is a kind of a transgression hypothesis, which I certainly discussed, which we found certain long non-coding RNAs that are probably transgressed by the host RNAs as well. And as a result, these long non-coding RNAs might really play a very important role towards viral host pathogenesis. But interestingly, as I told you, this long non-coding RNA is known to be highly expressed in prostate. So there is a plausible correlation between the patients with cancer prostate victims where their mortality is comparatively less when compared to the non-cancer prostate victims, during the COVID times. Okay. So the third part, the very important question, like any systems biologist would ask, is can we come up with a predictive analytics? So now that we have some huge chunks of data, so we have some data that is already existing. So we have the whole exome data, and we are presumably getting the whole transcriptome data. And we have some other consortium data that is publicly available as well. So can we apply some predictive analytics? Can we use some machine learning heuristics to decipher the knowledge and say that, say from 500 exomes, we have these many variants taken into account these pathodiagnostics? Can we really come to a conclusion, say, from 501st patient sample, we will not probably do the whole exome sequencing. Probably we'll be ready making up some kind of a panel, an NGS-based panel or a biomarker-based panel, where we will subjectively use these particular predictions and probably ensure that the clinician is given a kind of an upper hand or a kind of an advice that he could plausibly have these kind of variants sitting in them. So this is what we are currently looking at. So the part one is there are whole exome sequencing data. There are some variants that we have. And in part two, we have the regulome data, which we call it as the regulatory elements constituting the long non-coding RNA-protein interaction data. And we are also having RNA-seq data. We have obtained these reads of a few samples. And as you could see, we have about 120 samples and about 200 prioritized SNPs, which means about 24,000 variants. And we have validated about 5% of it so far. And we need to go for a precision and recall. So there is a good algorithm called deep variant, which has been used for breast cancer as well. It's just like Spotfire, the tool that probably many clinicians would use to infer where exactly this particular tumor is seen. And but this particular data, what we are trying to do is not the image-based data, but a kind of a binary data based on these particular interactions and the variants that we have. So the very first question that we asked towards this is, if at all there are certain variants, are these particular variants or the genes harboring these in nature? And if at all, if they're regulatory, can we say that these regulatory elements could really play a very important role towards the pathogenesis? And that's where, as I told you about the CTC of motifs, there could even be the DHS, so the DNA is one hypersensitivity sites, the transcription factor binding sites, the enhancers, the regulatory build molecules, which play a very important role towards pathogenesis of these particular things. Our end goal is to come up with a kind of a product. So this product should or need not necessarily be specific to in the Indian subpopulation, but it could be globally as well. But if you could see here, from the diagnostic front, about there are different cost remedy based approaches. Say, for example, for a PSA, it would cost this much. For digital rectal examination, it would be this. For TRUS, it would be something like this. For biopsies, it's a whopping up to 2,650 as far as 2017 records. So, which means in Europe or in India, it's fine, but in Asia or in third world countries, not all people would probably have, health insurance. And absolutely, come up with a very important question. Can we come up with a kind of a small pregnancy-like kit? Can probably a person who is aged above 55 with a scrotal pain, with a stigma in him where he cannot really express even to his children, can he probably have a small urine-based test, just like a pregnancy-based test, where it will probably detect, prostate, whether or not, he has a prostate cancer or not. So that is what, at the end, we are just, trying to come up with. So that's where this next generation sequencing data with long non-coding RNAs as divisive markers we are trying to explore. So the advantage here is there's absolutely a kind of a high turnaround time. Of course, we need huge computational space and good machine learning based algorithms, which we have in the form of deep variant, which we are trying to benchmark. And of course, the viability is also very important, apart from other proliferative measures, which probably we might get from the electronic health records, from the clinic. So
Prash, connection is lost. Prash, do you hear me? Connection is lost. Prash, do you hear me? I hope he will be back soon. You can state your questions using the chat, or after the presentation, you could use your micros and ask Prash about any unclear questions, or you would like to know more about his work, and I hope he will be back soon. Please use your chat if you have any questions. I have received, yeah, Prash, are you here? Yeah,
I'm so sorry. I apologize for this, for this, connection
connection,
problems.
yeah.
Yeah.
Okay, please
So,
continue.
yeah, I'm almost done with this. Yeah, I'm in the last slide, I guess, last or two slides. Yeah. So, um, yeah. So, yeah. So, this is where, we are trying to look into that. And the present results, support, a kind of a multi-gene, panel testing, which we are trying to look into that. So we'll have probably an admixture of, coding in as well as non-coding SNPs, and of course the phenotypes associated with that. And of course, we'll also look into the Gleason score attributes. And of course, the distant goal is, which I really don't see anything happening during the next 10 years, would be hereditary cancer prostate, where the US has already started. And of course, we'll also consider these particular informative predictors, for other association approaches. So future directions is, we probably do not want to start with cancer prostate itself, but we might want to ask the other way around. So what if there is a person who is really obese, who is aged above 55? Can he probably, would he likely get this particular cancer prostate during his lifetime? Okay, so these are the kind of questions that we are trying to ask, and we have identified, perhaps the very first time looking into the Indian specific variants. So not only the diet, not only the lifestyle, not only the obesity based things, but also the vitamin D associated elements are associated. There are also other point mutations which we need to look into this, because the whole exome sequencing as a fallacy, where we'll not probably identify certain copy number variants or single copy number variants. And that's the reason why we are looking for the RNA-seq based approach, and we'll also employ nanostring, which is a method, that would probably work, something related to digital PCR. And in the near future, this will also set a kind of a benchmark where will not only categorize only the cancer prostate or the malignant tumors, but there might also be a chance that the indolent tumors like the benign prostatic hyperplasia, or probably the persons with fistula or hemorrhoids or piles, would they probably have a kind of a risk of cancer prostate in the future, which is not really known. There are certain cases, but from the clinician perspective, it is not really known. So I thank all my collaborators. The most important thing is my PhD fellows, Nidhi Shukla and Ayam Gupta. Nidhi is a women's scientist fellow, and as well as my first PhD fellow for this particular cancer prostate project. Ayam Gupta is working on the transcriptome genetics for vitamin K and as well as D and cancer prostate. Shalini Rajgopal, Anchita Guman, Harshita, Sonal, and others are my other PhD fellows. But most of these particular collaborations come from a major funding called BrainPan, which is an electronics health records company, where they have really funded this particular project. And my clinicians, Devendra Sharma and Manish Vijay, both are urologists and pathologists respectively. And these particular people. And we're also a part of a practical consortium, which is an Australian-based consortium, which is co-led by Jyotsna Batra, who is a cancer prostate leader. So thank you, Ira, for this wonderful invitation. I look forward for interactions. Thank you so much.
Yeah, Presh, thank you very much for your excellent presentation. Very interesting and closely related to our research interests as well. And do you have any questions? And as I said, you can use the chat, or you can use your micros to state the questions. I don't see Any questions in chat?
So if there are no questions, you may as well reach me at these email addresses. So
Yes,
you're most welcome.
Yes, yeah, you have provided your email addresses, and which one is the best to contact you? The first one or the second one?
Yeah. The first one, yeah.
Okay. Yeah, Prash, again, thank you very much for your excellent presentation. I have some questions, but probably I will state these questions using email because this is a long discussion, and probably it is not necessary to discuss it now. I hope that we could also collaborate in future, and we will stay in touch. Thank you very much again. And bye.
Yeah. Thank you, Professor Scott. Yeah, thank you so much.
Yeah.
Thank you. Bye-bye.
Bye. Bye.
Thank you very much.
Thank you. Bye-bye.